Sorry, there is the chat. Please keep, yes. Thank you so much. Yeah, thank you so much, Raghav. All right, sir. Over to you, sir.
Thank you very much, Sri Gauri, and welcome to all of you. Let me set up my screen share. Okay, just a second. It's taking a little bit while to set it up. So before that, while I set up, can I have, or did you already cover this? Can I have a an idea? How many of you are familiar with microbiome or metagenomics data analysis? Just if you know this, just rank yourself from 0 to 5, and then type a number here. 0 would be complete newbie, and 4 or 5 would be somebody who can read through the papers or that contains analysis of metagenomics data, and then understand what they try to do and how they have approached the process. So let me set up this screen until then. So please put it in the chat box. Following this, I think now we are good to go. 3 basics, 0, 0. Thank you very much. Okay, thank you. So we have 0, 0, 3 basic. I would give myself 1 out of 5. Reading the information is where I'm struggling at. Okay, we will go through a lot of these methods. So, okay, that gives a good idea about how we are going to proceed in this session. So I'm going to be going a little bit slower than usual, so that you can get a sense or you can get an understanding of what we are actually trying to focus on this, or what we are trying to measure, right? So, so I am Raghavendran, and I am a bioinformatics mentor and expert mentor with Pine Biotech. So I welcome all of you to today's session in this hackathon series. So let's discuss. Thank you very much, everybody, for your responses. So in today's session, we will discuss some basics about, for example, what is metagenomics, which is, in simply put, is a way to study and explore microbiome. And how is metagenomics different from other types of omics data type and other types of omics analysis? And we will also talk about how the data is prepared, processed, and analyzed for metagenomics data. And we will also, again, discuss how it can be interpreted and what does it actually describe. So we will talk a little bit more about practical data and hands-on data analysis in the next session of mine. But before that, let's get an overview or understanding of what we are, why are we actually doing these, or why are we focusing on the metagenomics part to study microbial population or to study microbial communities, right? So to give you a sense or to give you a purpose, so to make you familiar with what is being studied and how are these studies and what are generally been, what is the general trend using this data type, right? And for most part, you know that we will be focusing on the bioinformatics part related to the metagenomics data analysis, and we will cover some of the main aspects of these metagenomics sequencing analysis of 16S rRNA and how these are actually applied to answer some of the key microbiology research questions using the data analysis. So before we begin, let's see how sequencing technology, or let's discuss how sequencing technology has helped us to study microorganisms and how these data analysis tools that are developed for metagenomic studies are used to characterize microbiota and ask some important questions about this microbial community, how these have expanded our approach or expanded our view to understand microbiology. So let's start from absolute basics. The interest in microorganisms around us has started since the discovery of microscope, and as the resolution actually improved, more and more members of these microorganisms like bacteria, fungi, and even viruses have been discovered, right? And at the time of development, the scale of this optical resolution can actually have reveal only up to 1 micrometer, which is a typical size of bacteria. But today, we know that microscopes are tremendously powerful. And this actually allows us to see smaller and smaller elements and reveal how the world of microorganisms function in every imaginable surface that is actively it actively interacts with each other. So in this picture, for example, or in this image, for example, we can see a super high resolution image that shows the process of bacteriophages in green attacking E. coli, that is bacteria, in blue. So what this actually shows us is not only that that there are microorganisms that are all around us in varying sizes and varying numbers, right? And they also tell us that they interact with each other. And these interaction will actually affect each other in various different ways. And why is this actually important? So what's important about this is the way that we previously study these microorganisms was through isolation, by isolating specific microbe or bacteria and growing it in the lab in culture medium, and then characterizing the microorganisms by different properties. And these studies have actually provided us important information about the function in isolation and the characteristics of these microbes in isolation. However, as we proceed in this fashion, more and more studies actually reveal that we know that many of these organisms, or most of these organisms, in fact, live in communities that are dependent on each other, communities that affect each other. And among these microbial communities, we will not be able to, or we could not culture many of them and grow all of these members of these communities in the lab. And in fact, majority of these microbes could not be cultured at all, or they are uncharacterized, right? And this is where sequencing efforts and sequencing methods actually began to shift the whole field as the development in sequencing methods increased, right? So when we reach the next generation sequencing methods or high throughput sequencing methods, soon this became a way to study not individual microbes or individual microorganisms, but actually the whole community of these microorganisms in different types of environment. And by studying this in their environment and in a community level, their properties and their characteristics could be associated with different types of conditions. For example, disease conditions, for example, metabolic abnormalities, and even sometimes drug sensitivities. So this is where the next generation sequencing has actually completely elevated the study of microbiology and microorganism characteristics to the next level. Soon, so many of these studies actually emerged, so the availability of such NGS data for metagenomics was huge. And this huge volume of publicly available data that are deposited by large projects, metagenomics projects, like the famous Human Microbiome Project, or the Earth Microbiome Project, or some organ-specific projects like the Oral Microbiome, and many others have actually helped us develop a detailed understanding that there are these distinct communities and they have specific properties, and these properties could be associated with different regions of our bodies, right? That itself was a big understanding and revelation in this field. So, for example, in this case, you can see that you will have distinct microbial community on your cheek compared to the one that is around your mouth, right? And these communities actually form stable environment, and that cannot be washed away by our daily domestic purposes, right? So inside each community, there is a balance of bacteria, fungi, and viruses that control these species abundance and activity. And this microbiome have specific impact, or they have purpose to protect the skin layer, right? In this case, for example, the purpose is also to interact with the tissue and to cause or to participate in the inflammatory responses to protect against pathogenic infection and etc. etc. So several studies continue to explore this microbial community. Over the year, primary focus was on the gut microbiome, while other distinct projects around several other organs like mouth, liver, skin, brain, vaginal, and lung microbiome were also being explored. So the main aim of such projects is to understand how community or how these microbial community differs between healthy individuals and people with deficiencies or abnormalities, right? Because of such studies, today health-related interventions seek to manipulate these microbial population. For example, change the proportions of gut microbiome with diverse products such as probiotics, drugs or therapies, food, wellness products, and even by changing your way of life, by giving some alterations to your way of life. So since 1990s, many of these microbiome studies were significantly advanced through major initiatives such as metagenomics of the human intestinal tract and subsequently the human microbiome projects, and several huge studies that have actually made possible through the advancements in the metagenomic sequencing itself, right? So the result of such studies and several ongoing studies is that we have skin, lung, gut, even brain microbiota characterized for the healthy individuals, and they are available for us to download and refer and to proceed with further analysis. So these studies and their data are extremely valuable resources for us to understand patterns among these microbial communities in association with different cases, or in association with chronic diseases, association with diabetes, obesity, or general well-being, etc. So there are several microbiome projects that were launched over time, and this is one of the popular field that got skyrocketed in the last 5 to 10 years. So among these several different projects, we have the Human Microbiome Project and Earth Microbiome Project are the largest projects in terms of data sets. There are other variety of projects like British Gut Project, American Gut Project, the Alzheimer's Gut Microbiome, and all these projects actually provide excellent account of gut microbiota for healthy individuals and Alzheimer's disease patients in this specific case, right? And a lot of them actually provide us with access to the data. And these data also, or these metagenomics data, these omics data, also come with some phenotypic information that could be clinical. And these data can also include a variety of factors like the activity of the individual, the diet of the individual, the medical history, their age, the BMI index, the ethnic and demographic background, etc., and other healthy habits like smoking, alcoholic abuse, and etc., Several of these phenotypic information are also related. And these factors could be explored for their association with the patterns that we could observe when we take data from these several projects and then start analyzing for our own projects, right? So there are some more super specialty projects like the cancer prevention gut microbiome, or the super interesting NASA's Gene Lab, which is a space-related omics database. And this database actually records omics data from either International Space Station to study the effect of radiation, or during space flight, or also they record data from controlled lab environment by mimicking the microgravity to resemble the space level. So all of these projects, and because of all of these projects and several other publications, today we know that the microbiome has a significant effect on the immune system, and it has this extremely important part to play in our ability to process specific food and extract different nutrients from food, and it also helps us with energy generation, with the metabolic regulation of the fat storage, and with processing of different types of environmental chemicals, and maintenance of different barriers and prevention of invasion, etc., etc. So when studying these microbial function or microbiome function, we are not talking about an environment that is completely separated from the host. But what are we actually considering, or what are we actually studying, is a complex community that is integrated into the host's several functional systems, right? And these communities could actually help and protect us, or could cause disruption in this environment, or could actually respond differently to the invasion of different pathogens. And today we know that, several endogenous microbial community actually contribute to several of these functions that we just discussed. So how are these studies actually carried out, and what are the major approaches for performing microbiome-based studies or metagenomic studies, right? There are two major approaches, or two main approaches for sequencing the actual microbial community through the amplification process. So what are we actually sequencing? In the first approach, we can actually use the 16S ribosomal RNA gene, and which has some hypervariable regions that with which we can characterize the microbial community through taxonomic classification. And this method, and what does this method give us? This method will actually help us build a survey of members of this microbial community under study. And this will help us understand their proportional abundance, like how many of these entities or how many of these members are present in what number, right? And they will be, they can be probed for their uniformity across different conditions, or uniformity across different sample types that we are actually trying to understand their pattern. So another approach which is widely followed is the whole metagenomics approach, or whole genome approach to the metagenomic sequencing, which will help us in assemble, or which will help us to assemble the genomes, full genomes of these microbial entity or microbial of these microbes completely from scratch. And while the 16S ribosomal RNA approach will help us place these microbes into known categories or into known taxonomies, but this approach, the whole metagenomics approach, will actually help us not just place this into known categories, but we can also end up identifying and characterizing some new members which are not characterized earlier. So this approach can also be useful in, for example, functional characterization of these new members based on where they are placed in the phylogenetic tree, and moreover, more studies or more approaches like meta-transcriptomics approach, which studies not only these members of this community, but also their functional activity, as I just said, right? And what do I mean by functional activity? So by probing specific genes and what they actually produce and how they are related to different biological pathways, we can understand the primary functions of these microbes in their environment. And this is a much more challenging approach, because of the simple fact that we do not know everything about these microbes, right? And we have to use, in the event of not finding a match or not finding a comparative reference to compare their functionality or functional activity, so we have to resort to comparative approaches like sequence-based comparative approaches to infer their function based on similarity scores and based on identity of these sequences. So another way to approach this studies is to use a technique called mass spectrometry, and this will actually measure what proteins or what metabolites are actually being produced by these microbes to help us characterize the impact of these proteins or the impact of these metabolites and the interaction of these metabolites with the host system. And all of these approaches will actually help us study, for example, how these microbes interact with the host and how these microbes actually interact with the pathogen that might end up changing the, for example, changing the changing the composition of the microbial community in general, right? So this is how we will approach these microbiome studies. So while performing or while studying about this metagenomics studies, right, unlike other omics data types or unlike other omics study, metagenomics does not study a single organism, right? But it actually studies the whole community at once. It is actually a snapshot of a collection of organisms directly in their natural environment. So what do I mean by that? As opposed to genomics, proteomics, or transcriptomics approaches, which is a study of an individual for different data types. And in metagenomics, we are actually studying about the data from a collection of organisms. And we will end up characterizing the community as a whole, as opposed to individual in the other omics data types. So thus, metagenomics approaches will actually enable the population analysis of previously unknown microbes, previously microbes that could not be cultured in culture media, right? This is extremely important, as the ability to identify microbes without a priori knowledge or without a prior knowledge of what the sample contains is actually elevating or is actually opening new possibilities or new doors in the disciplines like virology, in the disciplines like microbial ecology, in biomedical research, in microbiology, in environmental sciences, and generally. So, and at the forefront of this development is the sequencing-based examination of the metagenome that has become extremely powerful tool for actually generating novel hypotheses that can be verified and that can be verified for their presence in the pattern that we are observing from these metagenomics data. So practically how we would approach this is when we look at the example, for example, in the microbial community in the guts, we can look at the microbial community in the guts by taking the fecal sample as representative of the gut environment, or we can also take a cecal sample tissue by scrubbing the intestines. And from these samples, we want to organize the proportions of specific types of microbes by comparing their specific sequences from our samples to the known types of bacteria in our database or in a database in general. This approach actually helps us identify the taxonomic classification of the members in our sample. To validate this, we could actually either perform classification of those fully assembled genomes to understand variation from the known genomes from the database, right? When we perform metagenomics whole genome sequencing approaches, we can actually take the fully assembled genome and compare it with the reference or known genomes from NCBI or other reference databases and identify the variation that is present in our assembled genome. This could actually lead to additional insights about the microbiome of our sample, and this could actually, for example, lead to understanding, for example, antimicrobial resistance that can be characterized over several or individual variants in the genome that we could be listing out or that we could be identifying from these comparative approaches. So these studies, actually through these studies, we describe taxonomic classification, genomic classification, and actually and even variant-based classification can be performed primarily with the help of next generation sequencing method or high throughput method. But of course, if you know what are the target species or target members of the microbiome that you want to quantify, then you can also use qPCR to quantify them. So about the next generation sequencing, we are primarily going to consider or talk about 16S amplicon metagenomics, but there are also additional what happened here? Yeah, but there are also additional types of metagenomic sequencing that are possible that we already discussed, like the whole genome metagenomic sequencing. So whenever we have the sequencing data, we need to align it to a reference genome, right? And so in the case of 16S amplicon or rRNA sequencing, instead of aligning it to the individual genome, we will use a database of similar shorter sequences annotated by the taxonomical rank, right? The primary database that we will use is the Silva database, where we have a classification of different types of commonly found 16S ribosomal RNA sequences across the bacteria, and we try to match them to those sequences from our samples. Right? And what's good about the 16S ribosomal RNA unit is the length of this gene is approximately 1500 nucleotides long, and this gene actually contains several hypervariable regions that vary in the level of conservation across different species, right? And these hypervariable regions provides us with the signatures that we can use for bacterial classification and identification also, right? And this is widely used in several of the microbiome studies, and such approach actually focuses on the 16S ribosomal RNA, and while, for example, there are approaches that will sequence the complete sequence of the 16S rRNA gene, but a more favorable approach is the amplicon sequencing method, where more than one hypervariable region can be combined together to bring the target sequences to approximately a length of 300 base pairs long. And this is very convenient, and this is very useful length, because the sequencing in the commonly used MiSeq or other sequencers, which will produce approximately 300 to 350 base pairs long reads. So the comparison between the reads that we are generating and the target base pairs or target sequences will be extremely straightforward and will be more reliable. So, so this is one of the popular approaches to study or to quantify the level or quantify the amount of different types of microbes present in our sample. So when you have a 16S rRNA amplicon sequencing data, the objective is that the sequence reads actually can still contain variation, right? And we don't know whether the reads that we have or the variation present in the reads that we have could be attributed to biological variation that is significant, or could be attributed to technical variation due to the noise of the data recording. So instead of looking at these reads as individual reads, which is usually the approach in other omics data types, we in metagenomics data analysis cluster these reads into several clusters. And these clusters can be known or called as operational taxonomic units, which are simply reads that are organized by a specific similarity level, right? And these individual OTUs could be identified by a marker position in them, right? For example, this OTU has a marker position at this specific position that is different from the other OTUs. And similarly, for the other OTUs, they are characterized by their specific marker position at different places. This is just an oversimplification, and in reality, we will have more than one marker position that is for several of these OTUs, right? And these could be assigned with the taxonomy by comparing these marker positions and by comparing generally the sequences of these OTUs to the database. So what happened here? Yeah, so this database, the primary database that we will consider is a Silva database, where Silva database has a comprehensive resource of all the 16S ribosomal RNA of all the bacteria. And we have other databases like the NCBI RefSeq or NCBI database in general. We have GreenGenes, we have ribosomal RDPs, right? We have all these databases in there. But most preferred databases for bacterial classification is the Silva database, because it is regularly curated and regularly updated, right? And due to the participation of various research labs from around the world. So we can use this classification to characterize, for example, microbial composition that is specific to specific regions on the human body. Yeah. So in the taxonomic rank, we know that it goes from species all the way to the kingdom, right? And the decision that we will have to make while performing this level of characterization is which step or which rank should we use, right? This is a tricky question, or this is a tricky proposition, because sometimes for many of these bacteria, and more than one of these levels would be left uncharacterized, and we may have to resort to try a major or a higher rank like a phyla level or at a class level to quantify this. Or sometimes for the kind of data that we are actually analyzing, we might have most of their taxonomic rank fulfilled or characterized. Then we can go to a higher resolution like family level or even a genus level. But practically, genus level and species level are going to be extremely difficult because of the difficulty that I mentioned. Several of these bacteria are still uncharacterized at their genus and species level. So we will know only about family for these bacteria, etc. So this taxonomic level of the choice of the taxonomic level to understand their proportions will be different for different data sets. For this specific data set, which actually characterizes the abundance levels of bacterial species from different parts of the body, it is sufficient, or it is actually more informative when we consider the phyla level. And what we see is, what we observe is the firmicutes are quite abundant across the body, right? But actinobacteria is very specific to skin and the stomach region, etc., right? And is rather much present in much lesser or lower quantity in or lower proportions rather in the other areas of the body. So of course, while using the Silva database and trying to quantify the microbial species into their respective bacterial taxonomy is a simplistic approach, because this does not give us all the members that is present in the microbial community, right? And we will have more members, like there is also viruses, and that is actually the characterization of the samples for the different viruses that could be present in their community is another emerging field, and we call that specific data or that specific analysis as virome-based analysis, right? And there is also fungi and other members of this community, which is commonly explored when we have samples from environmental samples like soil or water or decaying tree material or plant microbiome, etc., etc. Right? So all of these projects that we actually discussed earlier, or I actually gave you some hints to, will provide us with the view of how these microbiome, or how the patterns in the proportional or abundance level of this microbiome could be associated with a variation that is either clinical in nature or either associated with particular diseases like Alzheimer's or cancers or any other diseases from or any other conditions like diabetes or like obesity, etc., etc. Right? Or in specific situations like in this case of development of microbiome in infants, right? And it turns out what plays a critical role in the early stages of this establishment of microbiome are actually multifaceted factors, right? Like these factors could include the food habits of the mother, and this because during the development of infant in the first 3 years, that is when their microbiome gets established, and this is actually several factors, as I mentioned, affects this establishment. The environmental factor, the food habits or general well-being habits of the mother, or even much earlier cases like risk during the pregnancy and their habits during the pregnancy. All of these actually will end up changing or will end up affecting the development of gut microbiota or generally other microbiota in the infants, right? While that is the situation for the infant, we can study, or we can, for example, relate to the infant's development of infant microbiome from the mother's clinical and well-being habits. For the adult microbiome, this is actually typically divided into several what we call as enterotypes, right? And which are primarily defined by diet and their location, and then they are influenced by lifestyle and culture and dietary habits and etc. etc. And what's actually interesting is that we have a view of healthy human microbiome by looking at the human microbiome projects, where we can have a lot of information about these kinds of lifestyle aspects, or these kinds of, for example, body mass index, and these kinds of where the person is actually located, and some other important insights like what are the general habits of these individuals, and etc. etc. Right? And some of the major hypotheses behind the gut microbiome and microbiome in general is that the established microbiome in any adult environment can become unbalanced, or can be disturbed by critical changes in the critical changes in the microbiome, or critical changes in one of the taxa being dominant, or one of the dominant taxa losing their dominance. We already saw that the presence of firmicutes being dominant over all of the regions in the human body. And for example, an imbalanced microbial population could be a deficiency in firmicutes, or the decreased or depleted levels of firmicutes in these or in any region, right? This could happen because of interaction between different attacks, or this could happen because of changing over time, right? Due to our food habits, due to our well-being habits, due to our lifestyle, like exercise, or like exposure to pollutants and etc. All of these can play a crucial role. And by understanding these patterns, we can make our hypotheses that can help us study the 16S ribosomal RNA type studies, and that will allow us to see the changes. And these changes can be interpreted, or these changes can be further developed to process and to develop interventions like antibiotics, or to develop interventions like probiotic, or to develop interventions like fecal implants, and etc. etc. Right? And these could be associated with, for example, a disease condition that are trying to link to those some specific outcomes. For example, if we can link the depletion or the dysbiosis is what we call the unbalanced state, right? And symbiosis is what we call for the balanced state. If we can link the dysbiosis to specific depletion or increase in the proportion of uncharacteristic bacterial member or phyla or genus, then we can start supplementing or changing the composition of these gut microbiota or other microbiota to the symbiosis stage, and hence we can find out whether this shift in the symbiosis or the shift in the balanced microbiota proportion will affect in changing the severity of the disease. And for some of the model systems, this has already been established that this is possible, and several of the research papers and are also available, and we in the OmicsLogic Learn portal have 4 specific projects or example projects that focuses on different levels of this association. We focus on, for example, taking the mouse model, or we study on taking the data from human gut microbiome, or we focus on combining inferences from one study and trying to find their echo, or trying to find their that pattern in another study, right? And more about practical approaches on all of these individual set in the next session, and where we will set up some of the analysis together and go over the outputs and go over the interpretations together. So these enterotypes that I mentioned have been associated with, for example, several of the dietary habits like high fiber fruits, or some vegetable type diets, or low fat diet, and etc. etc. And enterotypes or that promote symbiosis are actually usually associated with high fiber fruit and balanced vegetable diets, and etc. And enterotypes that are associated with promoting dysbiosis are more often than not associated with high fermented diet, or high cholesterol based diet, and etc. etc. Right? There are several of these patterns that is actively explored and also reported in several publications. And these enterotypes may also be affected due to the medicinal or due to the impact of other medicines that we intake, right? Not necessarily be antibacterial or antibiotic, but several of the non-antibiotic drugs also have an impact in changing the gut microbiome, or changing the enterotypes, or changing the dysbiosis, changing the balancedness of these proportions in our gut environment. This is also very well established studies, and there are also this is also linked in one of our projects. So the imbalance that we were discussing about in the gut microbial diversity is related to many modulations in, for example, infectious diseases and their pathogenesis. Many more studies, such as the Human Microbiome Project and data studies, actually uses the data from these human microbiome projects and follows their association in the role of this gut microbiome in, for example, Crohn's disease, for example, in the ulcer, in esophageal cancer, for example. They can also, there are several other studies that actually investigates the role of skin microbiome in psoriasis, in atopic dermatitis, in immunodeficiency, and they also try to relate or try to understand the role of urogenital microbiome in the reproductive complications. And as we already discussed, there are also a number of childhood disorders, including pediatric abdominal pain, including intestinal inflammation, and which is an extremely severe condition in premature infants, where the intestine in infants actually dies. So these can also be related to changes or in the dysbiosis of the gut microbiome of infant in this case, and for the adults in the other cases. Right? So how are these enterotypes that we just discussed, right, that we just spoke about, was actually discovered? They were actually discovered by studying samples from a variety of regions based on their difference in diets, based on their difference in ethnic background, based on their difference in the type of environmental exposure that they are exposed to, right? With this map, you can see that it gives us a sense of what types of bacteria that you can see that are specific to specific regions, and thus that gave rise to the idea of these enterotypes that are specific to specific responses from food habits and general well-being and lifestyle habits, etc., etc. right? And in addition to the microbiome, so far, majority of our focus has been with bacterial population. And that is where majority of the focus and many different studies have been published. And we have several, for example, evidences and results that actually links the symbiosis and dysbiosis state to the altered condition and imbalance condition in ranging from immunodeficiency to all the way up to neurological disorders. In addition to this, we can also look at viruses, which could be major part of this microbiome, as there are bacteriophages that are present in this microbial community that primarily interact with the bacteria, right? And we know that viruses can have major impact on several of the human diseases. And so they are also affected by this change in the proportions and may actually end up changing the or having an impact on the interaction of this microbial community with their hosts. So it is an interesting new field that is emerging, that is looking for the association of virome, right, for digestion of different medicine, and general association of microbiome for their responses to presence of different viruses that could cause actually disease, or sometimes how the microbiome could be protective against some of the viruses, but not care about or could actually allow some of the other viruses by not protecting the gut and the blood barrier through which these viruses enter into our blood circulation channel. So these are the different impacts. And for example, with that, I would actually like to conclude and pass it back to Sri Gauri or Spurs. And for the next session, we are going to talk a lot about practical approaches that we will cover in this. Okay, let's go back. Practical approaches that includes application of amplicon sequencing using several of the popular analysis methods like data2, KIME, and etc. So for the next session involves me, which is if one of you can provide us the date, we will actually look at how to set up this analysis practically in the tBioInfo server, and what are the parameters that needs to be considered, and how to extract data from publicly available resources like NCBI or resources from publication, and how to get hold of resources that we have actually identified and tabulated, and that could be helpful for you for the projects that you are focusing on. So if you have any question, I will be happy to take that.
Thank
Please,
you so
I
much.
think they can unmute themselves and ask and go ahead for the discussion. No?
Yes, sir, absolutely. So if the participants have any queries, please, you could either unmute yourself or you can put it in the chat and I'll take up the query. Does anyone have any queries?
Yes, I think we have someone raise their hand. Yes, please. Yes, okay. The question is, to predict, can we use AI tools? And what to predict? To predict which one? I mean, at the basis, I will approach this question with what are the other possibilities. The prediction that I was talking to you is about functionally annotating these microbial members in their community. So to do that, right? Exactly, right? Yeah. To employ AI tools for that, so we have to use some sort of structured data, right? So we may actually use the sequence to do that, but that, or if we can spend some time, we can convert these data, right, to structured data, right? And these sequence level similarities is also a structural, or is also a numerical entity that can be leveraged, or the sequence themselves can be leveraged into a long feature. And for example, the sequential composition for the GC level, or for different for different percentage of A, T, G, and C, and they also can be leveraged. So we can use feature engineering to generate a lot of features based on these whole genome sequences that are assembled for the uncharacterized part, right? But it can be done theoretically. It can be done, but I don't know whether such study exists or not. But it will be interesting to see how this can improve on the existing methods, like using phylogenetics or phylogenetic reconstruction based method, or phylogenetic tree based methods to characterize them, right? And I actually, I'll be interested to see what AI can do with this kind of data, right? But while coming down the path, right, AI is regularly employed where in places or in situations where we take these structured data, or the data that we characterize from these communities, and we can integrate them with the metabolomics data. We can integrate them with the metatranscriptomics data. So we can integrate multiple levels of these metagenomics and metatranscriptomics and metabolomics data to understand or to use AI for several other cases. Like, we can take these situations from healthy individual and from the individuals with some disorders, and we can try to use AI for that case. So that at the integration level, we can definitely use machine learning and artificial intelligence. Yes, Dr. Roki.
Hello, and thank you very much for this great lecture. I thoroughly enjoyed it. I remember there was a slide you talked about the different omics technologies that we use in Mics now, and while this hackathon is on metagenomics, there are the other omics technologies like the metatranscriptomics and up to metabolomics, based methods. So my question is this: would there be any order in terms of if you want to use any of these techniques combined in carrying out analysis, or would you select one based on what you're interested in looking for?
I mean, we can start actually with the metagenomics data, right? The primary difficulty in incorporating meta or taking data from metatranscriptomics or from metabolomics data is one that I already mentioned in the slide, that we don't know a lot of information about these, right? Many of these will have to be speculation based on comparative analysis, one. And two, the data is really huge, even when you compare it with the genomics or metagenomics data, right?
Yes, it will be a lot of data.
Exactly. The mass spec data is huge. So in that instance, what is preferred approach is we can take the processed data from these metabolomics or metatranscriptomics approaches, and our primary data could be from the metagenomics data, could be the abundance table that we are characterizing or that we are obtaining from the amplicon sequencing data. And in addition to that, over that, we can try to, as I mentioned, we can try to integrate these metatranscriptomics or metabolomics data to functionally understand the importance of different different bacterial or microbial entity. So at that level, we can definitely use the processed metabolomics and metatranscriptomics data.
Thank you very much.
Yeah, hope I answered your question. But
yes,
but the raw sequencing processing is actually the problem is multifaceted. One is huge. The data is really huge. And two, the methods are not as standardized as we will see for the amplicon sequencing data and the abundance level calculation data. So right now, for the abundance calculation data, we have two methods like DADA2 and KIME2, which is gold standard. You will not falter if you use those methods. But for understanding the effect of metatranscriptomics and metabolomics, several different research labs and research approaches actually use different methods and different approaches. So we have to spend a lot of time in actually in finalizing the approach and methodology rather than performing the analysis and trying to find implications and trying to find patterns. So that is one of the major hurdles in this case.
Thank you very much.
Cheers.
Thank you so much. So does the participants have any further queries? If so, please share them right now. And in the meantime, I would also like to remind all the participants that you need to create an account on the OmicsLogic Learn portal, as we will be making use of the resources on the portal as we have the sessions during the workshop. And please make sure that when you create an account on the portal, you need to update it as well, because the community manager and the mentors of the program will be viewing your account as well. So it has to look presentable. So please make sure to update it with your profile image, and your name has to be in proper capitalization, link to your social media handles, and a brief bio about yourself with your educational background and research interest. And also, if you're facing any technical issues while signing up or have any queries, you can mail your queries to Ayushi Notra. I'm sharing her email ID in the chat box. So please get in touch if you're facing any kind of issues. And also, please make sure to join the Skype group that we have for the hackathon participants, where you can share your queries that you might be facing. All right, and with that, if the participants have no further queries, I would also like to remind you that tomorrow we have the next session, and that will be by Dr. Shibi Chakravarti Kannan, and he will be speaking on the topic characterization of the gut microbiota in individuals with overweight or obesity during a real-world weight loss dietary program, a focus on the bacterioids 2 enterotype. So please make sure to attend the session as well and make the most out of it. And with that, thank you so much for today's participation, and have a great day. Thank you so much.